Okay, so welcome back to this next video in which we are discussing depression and antidepressant drugs. Okay, so we're now discussing the final drug that we're going to talk about, which is lithium, okay, which is a mood stabilizer used to treat bipolar disorder, and it is effective not just at removing the depressive periods, but also removing the manic periods, okay, and calming your mood down towards euphemia. Okay, so to understand how lithium works, okay, and I'm certainly not going to actually be able to answer how it stabilizes your mood, okay, but I am going to give you a pathway which we know that lithium acts on, which is potentially involved in how it acts as a mood stabilizer, okay. Now, it's going to interfere hugely with the GQ-mediated pathway. So firstly, we need to have a good understanding of the GQ-mediated pathway. And the GQ-mediated pathway is going to be activated by GPCRs, G-protein coupled receptors, hence why we have drawn the G-protein coupled receptor here. So, we've illustrated the key components of a G-protein coupled receptor. Now, all the G-protein coupled receptors, and I've already said that there's over 800 in humans. Whoops, what am I doing? I was about to write 880 there. Over 800, okay, in humans. Okay, all of them have these same basic principles. Okay, they all have these same basic structures that we've got here. Okay, right. Now, when the ligand molecule comes and binds to a G-protein coupled receptor, and all the different 800 G-protein coupled receptors will have different ligand molecules. Okay, I mean, some of them will share the same ligand molecule. For instance, I've already said that there are 13 G-protein coupled receptors for serotonin alone. Okay, but you know, there are many different ligands for different G-protein coupled receptors. Okay, what will then happen is the G-protein coupled receptor will change conformation so that it can interact with a heterotrimeric G-protein. So let me now describe to you what a heterotrimeric G-protein is. Okay, so once again, let these two parallel lines that I've drawn here represent the inner leaflet of the phospholipid bilayer, okay, which is the inner layer of phospholipids, and the outer leaflet of the phospholipid bilayer. So I'll just put that down. So this here, this is the inner leaflet, okay, and this here, this is the outer leaflet of the phospholipid bilayer. Okay, so the inner leaflet faces into the cytoplasm, and the outer leaflet faces out towards the extracellular fluid. Okay, so we now want to discuss heterotrimeric G-proteins. Okay, so this word heterotrimeric, firstly, okay, heterotrimeric. Hetero means different, okay, and trimeric means three-membered structure. So a heterotrimeric G-protein is going to be built out of 3 subunits, and each of these subunits is going to be of a different type. Okay, right. So the first subunit of a heterotrimeric G-protein, and arguably the most important, is the alpha subunit of a heterotrimeric G-protein. Okay, so I'll put this here. So this is going to be our alpha subunit of our heterotrimeric G-protein. Okay, now, alpha subunits of heterotrimeric G-proteins are special, okay, because they can be in 2 states. Okay, they can be in an on state, okay, in which they have guanosine triphosphate, GTP, bound to them, okay, and they can also be in an off state in which they have guanosine diphosphate, and I'll just get rid of that. Oh no, smudged it all over. Okay, and they can be in the off state where they have guanosine diphosphate bound to them, GDP. Okay, now, we will start with this alpha subunit in the off state, and therefore it's going to have guanosine diphosphate bound to it here. Okay, I'm going to colour it also in red here. So this is our alpha subunit of our heterotrimeric G-protein in the off state. Okay, now, the other thing to say about the alpha subunit of the heterotrimeric G-protein is that they have lipid moieties attached to them. Okay, at least one lipid moiety attached to them, which is going to anchor them to the inner leaflet of the phospholipid bilayer. So what is the principle here? Okay, the principle is that you are going to attach on a very hydrophobic molecule, and I'll give you an example of a very hydrophobic molecule that can be attached onto many alpha subunits. So many alpha subunits get palmitic acid attached to them. Okay, so what's the structure of palmitic acid? Well, palmitic acid is a molecule that would now be called hexadecanoic acid. So palmitic acid is the old name for the molecule that would now be called hexadecanoic acid. And although hexadecanoic acid is a bit more of a mouthful than palmitic acid, it's a more useful name because it tells us exactly what the structure of this thing is. It tells us that we're dealing with a 16-carbon fully saturated carboxylic acid. So here's the carboxylic acid group. Okay, now we're going to need 16 carbons. So here's the first carbon. So we're going to need 15 more. Okay, that means that 14 of these carbons are going to be in the form of methylene groups. Now, because I don't want to have to draw out 14 methylene groups, what I'm going to do is draw one methylene group. Okay, I'm going to put brackets around it like so. And then I'm going to subscript that 14. Okay, and what this means is take this methylene group here and copy it out 14 times. Okay, then after the final methylene group, then put a methyl group to finish the molecule off. Okay, so this molecule here, this is the structure of palmitic acid, which is also known as hexadecanoic acid. Okay, right. So what you will notice about this molecule is that it has this extremely long tail. Okay, and that this tail is extremely hydrophobic. It doesn't interact well with water. So basically, this is a lipid molecule. Okay, so lipid molecules are molecules with large hydrophobic portions, which this certainly has. But in particular, this is a special type of lipid molecule known as a fatty acid. Okay, right, so the basic principle here is that you're going to attach onto the alpha subunit of your heterotrimeric G protein a lipid molecule such as palmitic acid, and most alpha subunits of heterotrimeric G proteins do get at least one palmitic acid molecule attached onto them. Okay, this is now going to stick off the side of the alpha subunit like so. Okay, and this is going to be interacting with water molecules unless you stick it into the cell membrane like so. Now, lipid molecules do not interact well at all with water molecules. Okay, so this is what's going to prevent the alpha subunit from just floating around in the cytoplasm, because if it was floating around in the cytoplasm, that lipid molecule would have to be interacting with water molecules. So what's going to end up happening is it's going to end up shoved into the inner leaflet of the phospholipid bilayer like this, so that water molecules don't have to interact with the lipid molecule. And you can't pull the alpha subunit out of there without expending energy, because when you pull the alpha subunit out, it means that water molecules are going to have to start interacting with this lipid molecule, and that's going to be energetically unfavorable. Okay, so it means that the alpha subunit effectively ends up attached to the inner leaflet of the phospholipid bilayer. Okay, but understand this, it's not an integral membrane protein, it's dangling underneath the cell membrane like so. Okay, right. Now, when alpha subunits of heterotrimeric G proteins are in the off state like so, with GDP bound to them, okay, they can associate with two more subunits. Okay, now the two other subunits of a heterotrimeric G protein, these never ever part from one another. Under all physiological circumstances that are known, they stay bound to one another. Okay, so the other two subunits of heterotrimeric G proteins are beta subunits, okay, here in blue, and gamma subunits, which I'll have in green. Okay, and every single beta subunit will be bound to a gamma subunit. Okay, so beta and gamma never ever part. Okay, and it's a good job for the beta subunit because the beta-gamma complex, as it's called, so because beta and gamma never ever part, they're collectively known as the beta-gamma complex, although they are separate polypeptides. Okay, and the reason it's a good job that the beta always has a gamma is that the gamma also has a lipid moiety attached onto it, which again anchors it to the inner leaflet of the phospholipid bilayer. So again, the gamma protein dangles underneath the cell membrane. Okay, the beta protein has no such lipid moiety. It only is attached to the inner leaflet of the phospholipid bilayer through the subunit. Okay, so providing the beta subunit is attached to the gamma subunit, it too will end up dangling underneath the cell membrane like so. But without being attached to the gamma subunit, then it doesn't attach to the inner leaflet of the phospholipid bilayer. But it doesn't need to be worried about that because it always will have a gamma subunit attached to it. Okay, so the beta-gamma complex 2 is dangling underneath the inner leaflet of the phospholipid bilayer. Okay, now if the alpha subunit is in the off state, it will bind to a beta-gamma complex like so, and you'll then have a full heterotrimeric G protein. Okay, right. Now, there are many different types of alpha subunits, there are many different types of beta subunit, there are many different types of gamma subunit. Okay, so let me tell you about the different types of alpha subunit, and the different types of beta subunit, and the different types of gamma subunit. Okay, so we'll start off with the different alpha subunits. Okay, so there are 21 known different alpha subunits of heterotrimeric G proteins. Okay, so there is not just one alpha subunit of a heterotrimeric G protein. Okay, there are 21 known alpha subunits. Okay, in addition, there is not quite 21 different genes for alpha subunits. Instead, there are 16 different genes for alpha subunits. Okay, so there aren't quite 21 different known, there is 21 different known alpha subunits, but there's not 21 different genes, there are 16 genes. Now this may appear as a slight anomaly until you remember that there is the phenomenon of some genes having multiple splice variants. Okay, so let me just remind you, and just in case you don't know, what splice variants actually are. Okay, so splice variants. So basically, let me remind you of the central dogma of biology. So if we have these two parallel lines representing a piece of double-stranded DNA now, okay, so it no longer represents the inner and outer leaflet of the phospholipid bilayer. Instead, it represents a piece of double-stranded DNA. Okay, then let's say that this portion of the double-stranded DNA here, okay, let's say that this is a gene. Okay, so this is a gene. It could be a gene for an alpha subunit of a heterotrimeric G protein, for instance. Okay, so what then happens is, in the gene there are two strands of DNA. Okay, one of these strands is going to actually be used by the RNA polymerase II enzyme to produce the piece of mRNA from. Okay, the strand that's actually going to be used by the RNA polymerase II enzyme to produce the mRNA is known as the coding strand. Okay, the strand that is then not going to be used by the RNA polymerase II enzyme to produce the mRNA is known as the non-coding strand. So let's say this strand here in turquoise is the non-coding strand, whilst the purple strand is the coding strand. Okay, right. So what will happen then, if we want to actually make the protein associated with this gene, RNA polymerase II enzyme will work its way along the coding strand, like so, and synthesize a piece of mRNA here in blue. Okay, so here is this piece of mRNA, and I'll pull it out here. Okay, right. Now, this piece of mRNA is not yet ready to go for a ribosome and be translated. It is what is known as a piece of pre-mRNA, and before it actually goes for a ribosome to be translated, it needs to go through post-transcriptional modifications. Okay, so by the way, the process of producing a piece of mRNA from the DNA is known as transcription. Okay, right. Now the most important post-transcriptional modification is going to be called splicing. Okay, now what does splicing involve? Well, basically, the reason this pre-mRNA is not ready to go for a ribosome yet is that there are portions of this pre-mRNA that are not supposed to be translated. Okay, turning to, there are certain portions which are supposed to be translated, and let me highlight these now by drawing boxes around them. So let's say that these three portions that I have boxed, and I think I'll even colour them in. Okay, so I'll box them and colour them in orange here. Let's say that these three orange boxed portions are actually supposed to be translated. Okay, the portions that are supposed to be translated are known as the exons. Okay, so we've got these three exons here. the portions in between the exons which are not supposed to be translated, one of which we've got here, another which we've got here, another here, and another here, these are known as introns. Okay, right, so the process of splicing involves cutting the introns out and then stitching the exons back together to produce a much smaller piece of mRNA known as the mature mRNA. So let me show that here. Okay, so here now we just have the 3 exons joined together, and this is known as the mature mRNA. And this mature mRNA is now ready to go for a ribosome and be translated into a protein. Okay, now, that's all well and good, but how does this give rise to different splice variants? Well, basically, in the case of some genes, when you produce the pre-mRNA from that coding strand of the gene, there will be multiple different ways that you can splice it. Okay, so there isn't just one way to splice it. There isn't just one type of mature mRNA you can produce from this pre-mRNA. There are multiple. Okay, so let me give you a very easy to understand example of this. Let's imagine that this final exon, exon number 3 here, is not actually compulsory. Okay, so you can either include it in the mature mRNA or you don't have to include it. If you include it, you get the full-length mature mRNA. If you don't include it, you'll get a slightly shorter mature mRNA. Okay, that means there are now 2 different ways that you can splice the pre-mRNA. Okay, because depending upon what you're feeling like, you can either include this 3rd exon here in the mature mRNA, or you don't have to include it. So there are 2 different possibilities for what mature mRNA you can produce from this pre-mRNA. So there are 2 different ways you can splice. These 2 pieces of mature mRNA will create different proteins. I mean, this one's going to be a longer protein than this one. Okay, and these different proteins that are now being created from the same gene are then called the different splice variants of that gene. Okay, so this is the concept of different splice variants. That basically, some pre-mRNAs can have more than one way in which they can be spliced, okay? And that means that if you're making loads of these pre-mRNAs, some of them will be spliced one way, others will be spliced the other way, and therefore you'll get a mixture of the 2 different proteins being produced. Okay, right. Now, often it's more complicated than this. Some pre-mRNAs will have a huge number of different ways that they can be spliced, and therefore those genes will be producing multiple different splice variants. Okay, so that's how 16 different genes for alpha subunits code for 21 different alpha subunits. Okay, right. We're not going to fuss, however, over the different splice variants. This number is continuously going up. I think it is actually outdated now. Okay, it was true in a review that I read that was from a while ago, okay? But there will be more splice variants. I'm sure there are more known alpha subunits now. But however, the number of genes has not changed. There are still only 16 genes for alpha subunits, even if the number of splice variants is going up. Okay, right. So I'm going to tell you the names of the 16 different genes for alpha subunits, and we're not going to dwell on the splice variants. Okay, to all extents and purposes, the different splice variants of the same gene have the same function anyway. Okay, right, so, oops, not onto that. Let me tell you about the different splice variants then of not the different splice variants, the different genes for alpha subunits of heterotrimeric G proteins. Okay, so, they are grouped into 4 different families. Okay, so family number 1 then is the G-alpha S family. So the way that you write alpha subunit of a heterotrimeric G protein is that you put a capital G like so, you subscript it alpha, and then you put a subscript on the alpha which tells you what type of alpha subunit it is. Okay, now this, we're not talking about a specific type here, we're talking about a family now. So there is the G-alpha S family of alpha subunits of heterotrimeric G proteins. Now this contains the G-alpha S gene itself. Now G-alpha S has multiple different splice variants, but we won't go there. Okay, and then it also consists of another gene as well, the G-alpha OLF gene for alpha subunits. Okay, and this only has one splice variant. Okay, so this family, the G-alpha S family, contains two separate genes, G-alpha S and G-alpha OLF. Now, all of the splice variants of G-alpha S and the one splice variant of G-alpha OLF, these alpha subunits all do pretty much the same thing, which is why they're grouped together. They all activate adenylyl cyclases. Now, G-alpha S is the more important one. You find that all over the place. You find this expressed in many different tissues. G-alpha OLF, it's expressed in certain places, such as the olfactory epithelium, okay, hence its name, okay, and where it's expressed, it seems to be that it just replaces G-alpha S, okay, so in the places where you find G-alpha OLF, such as the olfactory epithelium, generally you find very low levels of G-alpha S, so it's as though G-alpha OLF has just replaced the G-alpha S in certain places. Okay, so those two are grouped together, so those are 2 out of the 16, and it makes sense that those are grouped together. Okay, in this G-alpha S family. The next family is the biggest and most difficult to remember. It's known as the G-alpha I/0 family of alpha subunits of heterotrimeric G proteins, and this contains 8 different genes. Okay, so firstly, it contains all of the G-alpha I alpha subunits. So it contains G-alpha I1, G-alpha I2. So people are often surprised when they find out this for the first time. For some reason, people think that there is only one G-alpha I alpha subunit, when in fact there are actually 3 of them. Now, they all pretty much do the same thing, so you'd be forgiven for thinking that there was only one of them. Okay, and often people will just write G-alpha I, okay, and what they'll mean is one of the 3, okay, because they all do pretty much the same thing, and often G protein coupled receptors which activate them, or one of them will activate all 3, okay, so they're very similar in structure. But they are 3 separate genes coding for separate proteins. Okay, right, so there's G-alpha I1, G-alpha I2, and G-alpha I3, okay, then there is the other one after which the whole family is named. There is G-alpha O. Again, G-alpha O has very similar structure to the 3 G-alpha I's and has pretty much the same function, that it inhibits adenylyl cyclases. Okay, right. Now, importantly, if you read papers, you'll hear them blabbering on about G-alpha OA, okay, and also G-alpha OB, okay. Now, these are not 2 separate genes, they are the 2 splice variants of G-alpha O. Okay, so I said we weren't going to dwell on the splice variants, but you can't read a paper without hearing people blabber on about G-alpha OA and G-alpha OB. Okay, so be aware that those are splice variants, not separate genes. Okay, so there is one G-alpha O gene, basically. Now we'll do the G-alpha T's, I think. In fact, actually, firstly, I think I'll do G-alpha Z, because G-alpha Z also has very similar functions to G-alpha I's and G-alpha O's. Okay, so these 5 all pretty much do the same thing. Now we're going on to ones that are quite different. Okay, there are the 2 G-alpha T's, so there is G-alpha T1 and there is G-alpha T2. Okay, now these 2 are involved in transducin. Okay, so basically, in the rod and cone cells in the eye, you have G protein coupled receptors that respond to light. Okay, so when light hits the G protein coupled receptor, it then becomes active, and it's going to activate a heterotrimeric G protein. Okay, now the heterotrimeric G protein that is found in rod and cone cells are called transducin G proteins. Okay, and they are heterotrimeric G proteins, and the alpha subunit either consists of G-alpha T1 or it consists of G-alpha T2. Okay, in rod cells, the alpha subunit of the transducin heterotrimeric G protein is G-alpha T1. In cone cells, the alpha subunit of the transducin heterotrimeric G protein is G-alpha T2. Okay, now, they do not inhibit adenylyl cyclase enzymes. Instead, they activate phosphodiesterase 6 enzyme, which then breaks down cyclic AMP, cyclic GMP. Okay, so these cause cyclic GMP to go down within the rod and cone cells. Okay, now why are they grouped together with these 5? Well, these 5, by inhibiting adenylyl cyclase enzymes, they cause cyclic AMP to go down. Okay, so these cause cyclic AMP to go down, these cause cyclic GMP to go down. Okay, so that's the link between them basically. They both cause cyclic nucleotides to go down. So that's why the G-alpha Ts are grouped with all of these ones which cause cyclic AMP to go down. Okay, right. Finally, there is also G-alpha GUST, which is involved in the gustatory system. So that takes us up now to 8 separate alpha subunits in this G-alpha I/0 family. Okay, right. So, the next family then is the G-alpha Q/11 family of alpha subunits of heterotrimeric G proteins. Okay, now, the most famous member then of the G-alpha Q/11 family is G-alpha Q itself. Okay, so that's easily the most famous member, but there are 3 other members which pretty much do the same thing as G-alpha Q, which are G-alpha 11, the other one after which the family is named, okay, then G-alpha 14, and then finally G-alpha 15/16. Okay, so these are 4 separate genes. Now, do not get confused with G-alpha 15/16. Do not think that that is more than one gene. Okay, and to make sure that you never think that it's more than one gene, I'm going to tell you the story behind how it became known as G-alpha 15/16. So basically, people working in mice discovered what they thought was a new alpha subunit of heterotrimeric G proteins, and they called it G-alpha 15. People then working in humans found what they thought was a new alpha subunit of heterotrimeric G proteins, and they called it G-alpha 16. Later it transpired that these 2 alpha subunits, one that we'd found in mice and one that we found in humans, were actually the same thing. Okay, they should be thought of as the same thing. This is the mouse equivalent of G-alpha 16, basically. So, there was a movement then to have both of them renamed, because we don't want it called one thing in mice and then called a different thing in humans. So we had a movement to rename it G-alpha 15/16. Okay, it hasn't really worked. People working in mice still call it G-alpha 15, and people working in humans still call it G-alpha 16. But it would be nice if you showed winning and used G-alpha 15/16. Okay, now all 4 of these pretty much do the same thing, and these are the 4 that we're going to be interested in. I said we were going to focus on G-alpha Q because that's the most famous one, but actually all of these other 3 do the same thing, which is that they're going to activate phospholipase C enzymes of the beta form. And in fact, G-alpha 11 is almost identical to G-alpha Q, and many of the G protein coupled receptors which activate G-alpha Q containing heterotrimeric G proteins will also activate G-alpha 11, G-alpha 14, and G-alpha 15/16 containing heterotrimeric G proteins. Okay, so that takes up to 14 genes now that we've seen. Okay. Now finally, there is the final family of alpha subunits of heterotrimeric G proteins, which is the G-alpha 12/13 family. Okay, and this consists of 2 members, the G-alpha 12 protein and also the G-alpha 13 protein. Okay, so these 2 are the ones that no one has ever heard of, and they haven't even heard of a single member in this family. Okay, these are involved in cytoskeletal remodeling, and I really am not going to say anything more than that. Okay, so that's our 16 different genes for alpha subunits of heterotrimeric G proteins. Okay, now, so that you have a complete picture, I'm going to tell you about all of the known beta and gamma subunits as well. However, the beta and gamma subunits, as far as we can tell, it does not matter at all what beta and what gamma subunit you have. in the heterotrimeric G protein as far as the signaling of that heterotrimeric G protein is concerned. All that matters is the alpha subunit. However, for a complete picture, to broaden your knowledge, I'm going to tell you about the beta subunits. Okay, so there are 5 genes for beta subunits. Okay, and these code for 6 known proteins. Okay, so that would have seemed a little bit suspicious, but we now know about splice variants. So we know that one of those genes must have 2 splice variants, and indeed one of those genes does have 2 splice variants. Okay, so the naming of beta subunits and indeed of gamma subunits is far simpler than it was for the alpha subunits. Okay, so you have G beta 1, okay, G beta 2, and I'm showing the genes at the moment, G beta 2, G beta 3, G beta 4, and G beta 5, which is just lovely. Okay, G beta 5. So, which of these 3 genes has 2 splice variants? Well, it's the G beta 5, okay, and the 2 splice variants are called the G beta 5 splice variant, okay, and then also the G beta 5L splice variant, okay. So those are the 2 separate splice variants of G beta 5. Now, all of the other G beta genes, they only have 1 known protein. Okay, so G beta 1 produces the G beta 1 protein, G beta 2 produces the G beta 2 protein, G beta 3 produces the G beta 3 protein, and G beta 4 produces the G beta 4 protein. Okay, it's just this one that you have to watch out for. Right, so that's the beta subunits, and now let's move on to the gamma subunits. In the case of the gamma subunits, there are 12 genes, okay, and then there are 12 proteins. So there are no splice variants in gamma subunits, so splice variants are not a big thing in the gamma subunits. Okay, now it's almost perfect with regards to the naming, but there is one little annoying blip in the naming of the gamma subunits of heterotrimeric G proteins. Okay, it goes up, it starts very nicely. There is G gamma 1, there is G gamma 2, there is G gamma 3, there is G gamma 4, okay, there is then G gamma 5, but there is no G gamma 6. You go straight past G gamma 6, you go on to G gamma 7, G gamma 8, G gamma 9, okay, G gamma 10, and then it's going to have to go up to now G gamma 13 rather than G gamma 12. So G gamma 11, G gamma 12, and G gamma 13. Okay, so those are the 12 different gamma subunits of heterotrimeric G proteins. Okay, right. So, we've now discussed all the alpha, all the beta, and all the gamma subunits. Now, it does not matter, as far as the function of the heterotrimeric G protein is concerned, which beta and which gamma subunit you have. All that matters is which alpha subunit you have. Okay, which is why heterotrimeric G proteins are named according to which alpha subunit they have. Okay, so for instance, you can name a heterotrimeric G protein the GQ heterotrimeric G protein, and what that means is that I don't care what the beta and the gamma subunit are, the alpha subunit is G alpha Q. Okay, right. Now, in reality, I'm going to put this little dash 11 there. I'm going to put GQ/11, okay? And what that really means is that it could be any of these 4 here, okay? We've got a heterotrimeric G protein where the alpha subunit is within the family of G alpha Q/11. It's one of these 4. This is the pathway we're going to be interested in. We're going to be interested in alpha subunits within this family, okay? So that's what GQ/11 like that means. It means a heterotrimeric G protein where the alpha subunit is within the G alpha Q/11 family. I'm not telling you specifically which one it is, okay, it could be any of them. Okay, but I don't care what the beta and the gamma subunits are. Right, so back to our other picture, okay, so basically G protein coupled receptors, every single G protein coupled receptor will interact with certain heterotrimeric G proteins, and the way it judges a heterotrimeric G protein is by the alpha subunit. It again doesn't care what the beta and the gamma complex are, okay? It just cares what the alpha subunit is. And each G protein coupled receptor, once active, will only interact with certain alpha subunits, so it's picky towards the G proteins that it's going to activate. It will only activate heterotrimeric G proteins with certain alpha subunits. Okay, so we're going to be interested in G protein coupled receptors which activate GQ/11 family heterotrimeric G proteins, which are those where the alpha subunit is in the G-alpha Q/11 family of alpha subunits. Okay, right, so we'll call it there for this video, and in the next video what we'll do is see what's going to happen once the G protein coupled receptor has been activated, how it's going to activate the heterotrimeric G protein, and how that's going to result in the activation of phospholipase C beta enzymes.